I started the recording. Okay. I'm going to divide chapter 9 up over 2 days since we're a little bit ahead. It gives me a little more time. On Wednesday, though, when I finish up chapter 9, I will move into chapter 10. I believe your next exam is about the 21st, I believe. A lot of biochem. Okay, so most, I believe, midterms are next week. But for y'all, it's bumped a little bit further down. All right, so chapter 9. We're here in the 5th of 7 traits we talked about that any organism must, or be considered a living organism, you must possess or exhibit these 7 common properties. And we're focusing on number 5, energy utilization, for chapters 8 through 10. Talked about last class how there's a total of 3 energy producing processes that fall under number 5, energy utilization: cellular respiration, fermentation, and photosynthesis. Talk about how these energy producing processes are sometimes referred to as anaerobic and aerobic metabolism, or anaerobic and aerobic chemical processes. But I said, despite that, okay, cellular respiration and fermentation are covered in chapter 9. Now we're going to spend most of our time on cellular respiration, just a brief moment on fermentation. But the reason for combining these two in the same chapter has nothing to do with whether or not they consist of a series of chemical reactions that somewhere along the line requires atmospheric oxygen. It has to do with the purpose. The reason why they place cellular respiration and fermentation, these are 2 energy producing processes in chapter 9, is because the purpose is to produce the type of chemical energy called ATP. Okay, and there are some other common features, but the key difference is cellular respiration consists of 3 stages. Each stage consists of a series of chemical reactions, so it's much more involved, much more tedious, much more in depth when we go over this. Whereas fermentation, it has multiple chemical processes going on, and it has one in particular that's synonymous with cellular respiration. But overall, it consists of a shorter number or fewer series or shorter number of chemical reactions that result in the production of ATP. And then we'll go into photosynthesis in chapter 10, because there for photosynthesis, the actual purpose is producing this type of chemical energy. Okay, it says a cycle of energy provided between plants and animals involves photosynthesis and cellular respiration, respectively. So here in chapter 9, this is the formula I want you to know. Now on the product side, there's 3 products produced by cellular respiration, this process. I put approximately, this means approximately here, 30, because it depends on the textbook you're looking. It can be as few as 30, as many as 40. In this textbook, I'll show you a figure towards the end of the chapter where they give you a set number, and that's fine. You can go off of it. But I usually tell students somewhere between 30 and 40, but I try to stay consistent for y'all. Okay, so approximately. This is the other chemical energy producing process we're going to cover in chapter 10, okay, called photosynthesis. Again, if you look over here to the right side of the arrow, you can see the product is not ATP. That is not the purpose of photosynthesis. It's glucose. But the reason why I say cycle of energy is because this is often how the 2 processes are related. Okay? What are the products for photosynthesis? Happen to be the reactants, okay, needed for cellular respiration. And then 2 of the 3 products, these 2 over here, produced through cellular respiration, happen to be inorganic compounds that are required for this other energy producing process, photosynthesis plus sunlight energy. So bottom line is you need to know both formulas, but as you're memorizing one, you're memorizing the other. Okay. When you memorize the formula for cellular respiration, these are the reactants. They also happen to be the products. Okay. And then if you look up here for photosynthesis, 2 of the 3 reactants happen to be 2 of the 3 products. So there's a relationship between the 2, a cyclical relationship between the 2. Now we're going to focus on cellular respiration, as I said. Cellular respiration requires a nutrient. The preferred nutrient by our cells is glucose. Our nervous system, it is glucose. That's it. So they show you here what we call a heterotroph, a consumer, consuming a plant, which is in this case an autotroph or photoautotroph or producer, consuming it for the nutrients that it needs. Now, not all nutrients, but some nutrients serves as a type of chemical energy that cells of our body can transform into a different type of chemical energy known as ATP through this process called cellular respiration. It's kind of like vehicles. You know, many still today run on gasoline, but some run on diesel, some like Tesla's basically electricity, some are hybrids. Well, proteins in our cells, the proteins in our cells that require energy to do work, the majority of them need energy in this form, ATP. Okay, so glucose is a nutrient that serves as chemical energy, but it just so happens most proteins in our cells need this type of energy. Now, we'll talk about a couple of exceptions towards the end of cellular respiration. Now, we also need an essential gas, no substitute, O2. So everybody's familiar with you inhale O2, you exhale CO2. What most people are not familiar with is everything that happens in between. The reason why we constantly need to breathe, okay, is because we constantly need O2. All right. The way it works is for every 1 molecule of glucose that a cell uses for this process called cellular respiration, if it has 6 molecules of O2, it's going to produce approximately this much ATP. However, if the O2 is not available, the cell can still start off using glucose for cellular respiration. However, it actually can't complete cellular respiration. It only completes stage 1, which is actually an anaerobic process. And instead of approximately this many molecules of ATP, the cell only gets 2. Therefore, the only cells that are not performing cellular respiration, which consists of 3 stages and involves these 2 reactants, are cells that are dead. Okay. Therefore, when you exercise to the point of fatigue, okay, that fatigue, you notice your respiratory rate picks up, your heart rate picks up, but eventually your muscular system says, that's it. We don't care what you want to do. We're not going to listen. We're not going to do anything. Well, your muscles start to shut down because there's a lack of oxygen. In other words, your muscular system goes from performing the function you want to pulling back and going into survival mode. Okay? They're trying to stop you and say, hey, look, we're going into survival mode. We don't have enough of this. Therefore, we can't produce enough of this to meet the demands you want. So we're actually going to force you to go take a huge step back. Well, when this happens and when both reactants are available and enough ATP is being produced, regardless of whether you reach the point of fatigue due to a lack of oxygen, as cellular respiration is occurring using these 2 reactants, we're always getting CO2 produced. Therefore, you're always exhaling CO2. So O2 you take in, primary function, cellular respiration. CO2 you're exhaling, it's coming from cellular respiration. Okay? Now, cellular respiration consists of 3 stages. Okay? One of the stages, as I'll point out to you, the very first stage, is considered a type of anaerobic metabolism. Okay? Our skeletal muscles can actually go from cellular respiration to a type of fermentation you're going to come to know as lactate or lactic acid fermentation. Okay? Go to producing ATP with the use of oxygen to producing ATP without using oxygen. However, they can only do it for a short while doing that type of fermentation. So for cellular respiration itself, even though one of the 3 stages is described as being anaerobic, cellular respiration actually consists of 3 stages. Therefore, it is considered aerobic. So make sure you know that going into the test. Cellular respiration consists of 3 stages, and therefore it is an aerobic process, or what they'll sometimes refer to as aerobic respiration. But you can put aerobic metabolism. I keep meaning to change that. Now, so it says here, cellular respiration includes both aerobic and anaerobic respiration or chemical reactions, but it's an aerobic process. Now, when cellular respiration is performed, carbohydrates are the preferred choice. That's the preferred nutrients such as glucose. However, fats such as fatty acids, proteins such as amino acids are also nutrients that can be used by a cell to produce ATP. But, okay, for our nervous system, it's always a carbohydrate. But for the rest of our body, it's preferred to have the option of using glucose because when the ATP is being made, the waste product, our body has been built to eliminate it. Therefore, there's no negative impact on our body long term, say over 10, 15, 20 years, like you see with the diabetic who can't use this. Okay. Now, our body will intentionally, you take an A&P class, okay, they'll talk about how our body will intentionally, on purpose, cause most of our cells to stop using this and conserve glucose for the nervous system and tell the rest of our body, hey, you can use the fats and proteins instead to produce energy, because short term it's great, but not long term, not 10, 15, 20 years, okay? Now, so listen carefully. Going into the test, the questions I test you on for cellular respiration, this is the formula you're going to feed off of. This is the only nutrient I'm going to focus for the next exam, glucose, okay? Now, this actually does not represent a chemical reaction that you see as a textbook chemical reaction in chemistry. Earlier in the semester, I gave you a chemical reaction. 2 atmospheric molecules of H2 plus 1 atmospheric molecule of O2 gives us 2 molecules of H2O. If you count up the number of hydrogens total and the number of oxygens total on the right side or on the left side or away from the arrow that are reactants, and you count up the number of hydrogen and oxygen atoms that make up the products or the product, you know you don't lose any atoms or gain any atoms. There's the same number of hydrogens and oxygens on both sides, okay? If it doesn't make sense what I'm saying to you, remember you were supposed to have had chem, okay? That's not the case here. This is more of a summary, okay? There are 2 reactants. The first reactant is also our substrate, okay? So glucose is also our substrate. Gosh, try not to curse. All right. I thought I had it. I lost it. Okay, so it's supposed to say substrate. So glucose is not only a reactant, it's our substrate. It's a nutrient that serves as chemical energy, but for cellular respiration, it is the substrate, okay, that is used. This is where it all begins with glucose here. We will talk about how cellular respiration is broken up into 3 stages. The first stage is glycolysis. The starting substrate is glucose. And then we will talk about there are a series of chemical reactions, 10 total, catalyzed by 10 enzymes that are focused on breaking down glucose by stripping electrons from it. Okay, you're going to have to know 2 of the enzymes or 2 of the 10 chemical reactions you're going to have to know. Okay, O2 doesn't come into play until stage 3. Oxygen is not a substrate, but it is a reactant. And it is extremely important, just as important as the nutrient. Sorry, that's ugly. It started off fine. Oxygen is described as the final electron acceptor, or you can say oxygen is a reactant that functions as the final electron acceptor in cellular respiration. Okay, so oxygen is a reactant that functions as the final electron acceptor in cellular respiration. So again, we have 2 reactants. One is a substrate. It's in the beginning. Everything starts with this substrate glucose. But then when we get into the 3rd stage, we will talk about how O2 is the final electron acceptor. And if it's not there, that jams up this whole process to where only stage 1 can happen, and we do not get near the amount of ATP a cell needs just to stay alive. Okay, you can see there are 3 products. The waste product is CO2. We eliminate simply by exhaling. 6 molecules of H2O. I got to fix it. I think it just got messed up uploading it, but 6 molecules of H2O, harmless. We're 2/3 water. And then the purpose of cellular respiration. Okay. Purpose of cellular respiration, to produce a type of chemical energy needed by proteins called ATP. Again, it says approximately 30, but it could be 30 to 40, anywhere in there. Okay. As I said, cellular respiration is broken up into 3 stages. 2 of the 3 stages, it'll be the 2nd and 3rd stage, happen in the mitochondria. So you have to know the anatomy of the mitochondria in order to understand what's going on as we move into stage 2 and especially stage 3. So just briefly, and I'll go over this again, but briefly, mitochondria has a membranous organelle, but it has 2 membranes, an outer membrane and an inner membrane that are separated by what is called an intermembrane space. The inner membrane here, okay, is folded, but what really stood out, okay, to scientists is what was inserted into this membrane. Transmembrane proteins that function as enzymes. It's studded, the inner membrane is just studded full. You can see here it's blanketed with membrane proteins called enzymes. These enzymes are involved in cellular respiration. You cut this inner membrane away so you can look at the inside, and the inside area is called the matrix. And you can see over here it says organic molecules and O2 will enter. Coming out will be carbon dioxide and ATP. Okay, now not all ATP, but the vast majority. So as I said before, cellular respiration is a three-stage process. These are the three stages: glycolysis, citric acid cycle, oxidative phosphorylation. Okay. Lysis means, excuse me, sorry. Lysis means to break. Glycolysis has to do with the breakdown of the substrate glucose. Okay. So glycolysis isn't going to occur. It doesn't matter if there's 10 enzymes ready to act on it. We have to have that substrate glucose based on the way I'm going to test you, everybody, okay? Glycolysis begins with the substrate glucose. What will happen is by the end of glycolysis, the final product produced by glycolysis is pyruvate. So we will reach a point as we go over glycolysis where I'm going to want you to know the first enzyme that catalyzes the first chemical reaction of glycolysis. When I give you the name of that enzyme, you should already know what the substrate is going to be, because I said it begins with glucose, the substrate. But then I'm going to give you the last enzyme out of catalyzes the final chemical reaction of glycolysis. I'm already telling you here, okay, that pyruvate's the final product of glycolysis. So when I give you the second enzyme, you should actually know the product. So you should know part of each, and I'll point it out some more as we go, okay? What happens then is you're going to hear me say glucose and glucose-derived products, okay? So if you hear me say pyruvate, that's one of two examples I emphasize as glucose-derived products. Now, we will talk about how once pyruvate is produced at the ends of glycolysis, it'll be transported into the mitochondria, it'll be converted into another molecule called acetyl-CoA that will then enter the citric acid cycle. Acetyl-CoA is the second example of a glucose-derived product. But the purpose of the citric acid cycle, it says here, is to complete the breakdown of glucose. Listen carefully, everybody, and I'm going to go over this again. The actual purpose of the first two of three stages of cellular respiration, it's not breaking down glucose per se, and it's not even ATP. It's taking or stripping electrons away from glucose and taking those electrons and transporting them to stage 3. So there are going to be some review questions I'm going to ask you, and hopefully y'all will do a hell of a lot better than the morning class, okay? Because it's review questions that come from chapters 2 and 5. So the purpose of these first two stages is actually to strip as many electrons from this organic compound glucose and transport them to this third stage, oxidative phosphorylation, okay? This third stage is responsible for the majority of ATP synthesis, and it consists of two substages. The first substage is known as electron transport, which I abbreviate this way. So we will talk about those electrons being stripped from glucose and glucose-derived products in stages 1 and 2. Those electrons are being carried, not just to stage 3, but specifically to the first substage of stage 3, which is electron transport. Then we'll talk about how there's a network of proteins that use the energy from electrons to help them do their job. You'll come to know this network of proteins as the electron transport chain. And we'll talk about how when that network of proteins does their job using electrons as a source of energy, it promotes the second substage, which is called chemiosmosis. And we'll talk about the star of the show for chemiosmosis. It's one of 5 enzymes you have to know going into the test, but this one's the easiest one to remember because the job of the enzyme is to synthesize ATP, the vast majority of ATP. So its name is simply ATP synthase, which I'll point out to you as we go. So this third step consists of 2 parts, and I'll explain where this long name, oxidative phosphorylation, I'll explain where this name, 2-part name, for the third stage is derived from these 2 substages here. Okay, so cellular respiration is a 3-stage process. The first stage, glycolysis, does not happen in the mitochondria. It happens in the cytosol or the cytoplasm. Okay, I usually say cytosol. It begins with the substrate glucose and it ends with 2 molecules of pyruvate. So they're showing you 1, but it's actually 2. I'll show you a figure I have that'll show you a molecule with a 6-carbon backbone and how when electrons are being stripped from glucose, it breaks it down to 2 molecules with a 3-carbon backbone that we call pyruvate, each of them. Once that pyruvate is made at the end of glycolysis, We'll talk about how it's transported into the mitochondria, and there's an intermediate step. I call it, in your notes, it's labeled intermediate step, but you'll come to writing yourself later that an intermediate step is called pyruvate oxidation. This is where this product from glycolysis now becomes a reactant and is converted to a different product, acetyl-CoA. Well, both of these, if you follow the arrows, both of these molecules came from that initial substrate, glucose. That's why I call pyruvate and acetyl-CoA glucose-derived products. They come from it. Then we'll talk about on Wednesday how acetyl-CoA is the substrate that is needed by the first enzyme that catalyzes a series of chemical reactions collectively called the citric acid cycle. Okay. Down here, if you notice, they show you there is some ATP that will be made during the first two stages, although they show you a little bit bigger figure here to indicate most is made here. But coming out of the top, notice something out of here and over here. Notice it says electrons. It says electrons are carried. Well, what did I tell you? The first two stages, the purpose is not actually to make ATP. It's to strip electrons from glucose and glucose-derived products and take them to stage 3, oxidative phosphorylation. Under oxidative phosphorylation, here's those two sub-steps or processes that occur: electron transport and chemiosmosis, which we'll visit on Wednesday. Okay. All three stages of cellular respiration consist of a series of chemical reactions or a chain of chemical reactions carried out by a group of enzymes. Okay, that's what this is telling you here. Okay, we talked about enzymes. Enzymes catalyze chemical reactions. They have an active site, which is a region that complements and binds substrate. Okay, and they're going to take a substrate and they're going to convert it into a product, and often they may need other how do you call it, molecules or substances to function as reactants to help them carry this process out. But this we haven't talked about. Okay, so bear with me, because this is what we call getting into a little bit of a hardcore biochem when nobody's usually had biochem coming into my class. It's rare. And even if they've had chem, it's not necessarily gone well. So if you look here, not only do enzymes catalyze a series of chemical reactions that make up all three stages of cellular respiration, but cellular respiration also involves the transfer of electrons and electron carriers. Okay, that's what you don't see in this formula. So right here it says electrons are extracted from glucose, okay, and transferred eventually to O2. So as we go through these three stages, we'll talk about how electrons are being stripped from glucose, which is breaking down glucose, eventually resulting in the production of pyruvate. Talk about how pyruvate will be transported in the mitochondria and converted to acetyl-CoA by stripping more electrons. Then we'll talk about how acetyl-CoA enters the citric acid cycle and more electrons are stripped to where we actually don't have a final product. In the citric acid cycle, there's no final product, so we'll talk about that more. But all these electrons that are being stripped from glucose and glucose-derived products are eventually taken to stage 3, specifically electron transport. Waiting at the end of electron transport is O2, our final electron acceptor. And what's been found is if it's not waiting there as a final electron acceptor at the end of substage 1 for stage 3, it screws up the whole system and we don't get the majority of this produced. So we'll talk about it more. What I haven't mentioned is how our electrons that initially are stripped from glucose eventually making their way to where they end up here at the final electron acceptor. It has to do with molecules called electron carriers. So electron carriers are molecules. I have a picture of at least one of the two that I'll show you because I had students start asking me. Some were sincere, but one day somebody I think was being something of a smartass. So I gave them the full molecule. I didn't get any more questions after that, okay? So you take biochem, it'll be different. They'll show you the full figure and they might tell you you need to know it, but listen carefully. Electrons stripped from glucose and glucose-derived products will be transferred to stage 3, where eventually they end up being accepted by O2. They will be transferred by molecules called electron carriers. There are types of electron carriers, which I'm showing you here and here. But I'm showing you these electron carriers, each electron carrier in two different forms. These boxes down here, for some reason on Collaborate, they're not where they're supposed to be. But I checked the PowerPoint I uploaded. These boxes are where they're supposed to be. So listen carefully. There are two electron carriers, and I'm giving them to you in two forms. One form we call the oxidized form. That's this right here, to the left of the arrow. Okay. If I'm talking to you about the two electron carriers, and I'm telling you, I'm talking to you about them in their oxidized form, one of the electron carriers is called NAD plus or positive. The other one is called FAD. Okay, just listen to that and I'll explain what oxidized means. All right. However, if these electron carriers are carrying electrons, specifically 2 electrons, the way a scientist lets you know is by rewriting them this way. Instead of writing NAD+, if I want you to know it's carrying or transferring electrons, 2 electrons specifically, I'll rewrite it as NADH. If the electron carrier FAD is gained 2 electrons or it's carrying 2 electrons, the way I'll let you know is I will rewrite it as FADH2. When you write them this way, the electron carriers, you're giving them in their reduced form. Now, what does oxidized and reduced mean? Has to do with down here. Cellular respiration is basically what they call a redox reaction. It consists of chemical reactions that are referred to as reduction and oxidation reactions. Whenever you hear that reduction and oxidation or reduced and oxidized in biochem, automatically tell yourself this has to do with the transfer of electrons. Okay, so if you look here, as we'll talk about in glycolysis, we'll talk about how glucose loses electrons. Enzymes strip electrons, okay, from glucose, but they say glucose loses electrons. Listen carefully. What's happening to glucose is it's being oxidized. But more generally speaking, not just glucose, but any molecule that loses electrons, we say it's oxidized. So when I write the electron carriers, these 2 electron carriers this way, I'm telling you they're oxidized. What I'm saying is they've lost or they're not carrying 2 electrons. Okay? But if you look here, I said O2 is the final electron acceptor. Well, sometimes what they'll say is at the end of electron transport, this process, O2 is reduced. What they mean is O2, okay, has gained electrons. Whenever, and more generally speaking, whenever O2 or any molecule gains electrons, we say it is reduced. Therefore, up here, NAD plus can carry up to 2 electrons. Okay, when it gains 2 electrons, we rewrite it in its reduced form, NADH. Writing it this way lets you know it's reduced, which means it's gained or is carrying 2 electrons. FAD, if it is reduced, that means it is gained or is carrying 2 electrons, but we write it out this way to let a person know, FADH2. So again, in general, any molecule that loses electrons or gives them up, we say it's oxidized. Any molecule that gains electrons, we say it's reduced. So again, these are oxidized electron carriers. These are reduced. You're going to see them a lot as we go through this. So we call them redox reactions. I'll let you read this. I'm not testing you on this stuff here. It's just in case. I have some students that take biochem, but just reread this. It's saying the same thing as I said before. And they call them redox reactions. Okay. So we're going to get into glycolysis right now. All right. We're going to talk about stage 1. This happens out in the cytosol. And glycolysis begins with the substrate glucose. We will talk about the first enzyme that catalyzes the first chemical reaction of glycolysis. And it's that enzyme that ensures glucose is the substrate that is used to begin glycolysis. Okay? The final products, not product, but product. Notice it says 1 glucose here, 2 pyruvate here. The final products of glycolysis are 2 molecules of pyruvate. Look at the chemical structure that I gave you. down below glucose and pyruvate. Some of the questions I asked this morning's class, I'm going to ask y'all. Glucose, inorganic or organic compound, everybody? There's 16 of you here, so we should get an answer.
Is it organic?
Right, easy enough. Okay, just use the chat board if you can. All right, listen carefully. What is the key atom that forms the backbone or skeleton of organic compounds? Right. What type of chemical bond does carbon like to form? Okay, thank you. Y'all did way better. All right, now there's the 7:30 a.m. So, a little bit in their defense, 7:30 a.m. Okay. What is the molecular formula, though, for glucose and any other monosaccharide? What's the molecular formula? Does anybody remember? Right, C6. See if I can do it. H12. But we said they're often referred to as structural isomers because even though all monosaccharides are made up of carbon, hydrogen, and oxygen. Ooh, that was terrible. I'll put the one over here. Let's see if I can do it. Nope, that didn't work. Y'all got the picture, okay? It's supposed to be, God, that was horrible. It's supposed to be a 12 down here. So they're structural isomers. You look at glucose and fructose, which the two of you were supposed to know. They both had the same molecular formula. Excuse this mess here. They're both supposed to be C6H12O6, but they're structural isomers because glucose is actually a cyclical molecule, has 6 sides, a hexagon. Fructose, even though it has the same molecular formula, okay, it looks more like it's a pentagon. It has 5 sides, okay? Now, so bottom line is glucose has 6 atoms of carbon forming its backbone. And I'm telling you, as this substrate, okay, is acted on by 10 different enzymes, we end up with the final product called pyruvate. We end up with 2 of them. And notice, each molecule of pyruvate has a 3-carbon backbone. Where did these carbons come from? Over here, glucose, okay? So I'll show you this. Now, glycolysis is the one of 3 stages that is described as anaerobic. Okay, it falls under anaerobic metabolism. There are 10 enzymes that catalyze a series of 10 chemical reactions. The first enzyme acts on this substrate. The last enzyme produces 2 of these, okay? And it doesn't require oxygen. However, we're talking about cellular respiration, and cellular respiration doesn't just consist of the first stage, glycolysis. It also consists of the second stage, citric acid cycle, and the third stage, oxidative phosphorylation. So overall, despite this, overall cellular respiration is aerobic because eventually O2 comes into play. But the reason why they point this out is because glycolysis is not only the first stage of cellular respiration, it's also the first stage of fermentation, which I'll show you. Okay, this looks really scary. You got to break it up into 3 blocks. Okay, remember what I said, glycolysis, the purpose is not to produce ATP. What did I say the purpose of glycolysis actually is, everybody? What is the actual purpose of glycolysis? Okay, Ian, if you are having trouble typing anything in, go ahead and turn on your mic when you need to, okay? Sorry, I didn't see your hand.
It's okay. Real fast, would you actually mind going back to the previous slide real fast to copy down something important that I might forget about?
What did you need to copy down?
The pyruvate molecule.
Yeah, these pyruvates, you can underline them and write products. They're the final products for glycolysis. Okay, so yes, everybody else, you're right. The purpose of glycolysis, to strip electrons or take electrons or simply oxidize glucose. You got it there, Ian?
I'm good now.
Okay. Yeah, slides are available online, but we'll slow it down a little bit. We'll have some time here. Not this course, 06. I was grading labs from another course, and it reminded me to appreciate when y'all are looking at the slides, even if they're available, okay? I'm happy y'all are looking, so just ask, okay? Because this other class didn't look at anything, so all right, that's another story. All right, so glycolysis begins with one molecule of glucose. That's our substrate, okay? And when it enters glycolysis, well, in order to make energy, you're probably going to have to use a little what? Energy. If you've ever worked out, you know this. You have to expend energy in hopes of, in the future, you will have more energy. So you work out. So it's a similar process at the cellular molecular level. When glucose enters glycolysis, it's going to take a couple of molecules of ATP to help with stripping those electrons. There are some enzymes that need it, not all of them, but some of the enzymes. And then one in particular, it actually needs ATP for two reasons, and I'll show you that because it's the first enzyme that catalyzes the process. So it requires some energy to help glycolysis occur, all right? Then as glucose continues through glycolysis, okay, what do we call it? The cell gets back what it used plus a little extra. So it requires two molecules of ATP to help perform glycolysis, but glycolysis produces 4 total, all right? Now, as the glucose continues to go through, why is all this needed and what's happening when this is produced? Well, the purpose. In the end, a total of 4 electrons will be stripped from what was once glucose. And these electrons will be split up in 2 pairs, and they will be transferred to the oxidized form of an electron carrier called NAD+, reducing each of those electron carriers to NADH. Okay, so a total of 4 electrons are taken from glucose, but 2 are given to one of these, and then 2 more are given to the other one, so we end up with 2 that are reduced. Now, by the time the process is over, we go from our substrate to our key products. Ignore the water for right now. We end up with our products. 2 molecules of 8. So as you're going through cellular respiration, tell yourself as you go from stage 1 through stage 2, keep telling yourself, I am tracking what happens to 1 molecule of glucose as it goes through glycolysis. And then I'm tracking any glucose-derived product as it continues to go through, okay, on to the next stage, citric acid cycle. This is what you're tracking. Now notice something at the bottom. You take these 2, and then you summarize what is the net effect of glycolysis. Well, the net effect of glycolysis says this. Glucose is an organic compound. It has a backbone made up of carbon, and carbon loves to form covalent bonds. And when you look at the overall chemical structure of this monosaccharide glucose, it's formed by covalent bonds. Therefore, when you strip electrons from this organic compound, what type of chemical bond are you probably going to break, everybody? If you're stripping electrons from an organic compound, what type of chemical bond are you probably going to break? Not a rhetorical question. If we have an organic compound and we're stripping electrons from it, no. What did we just say? I asked y'all a question. What forms a backbone? Carbon. What type of chemical bonds does carbon like to form? Covalent. So if we're stripping electrons, what type of chemical bond are we probably going to break? Everybody try to use a chat board. I'm real bad with the hand thing. I don't know why. Cheyenne, what's your question? You can turn on your mic if you need to. But the small p with the circle, that's a phosphate group. I don't know if y'all recall when we were going over functional groups. I told you sometimes for a phosphate group, they actually give you a capital P with the circle. And I was going to tell y'all that later anyway. Okay? All right, so for now, though, ignore this. I'll go over this more later. Listen carefully. It's an organic compound. It's not only carbon that loves to form covalent bonds, but all the other atoms, hydrogen and oxygen, are also forming covalent bonds, connecting all of them. So when you strip electrons from an organic compound, it's pretty much a guarantee you're going to break covalent bonds. So we go from a molecule with a 6-carbon backbone to 2 molecules with a 3-carbon backbone. That's what they're showing you here. What causes glucose to break down to 2 molecules of pyruvate? Electrons are being stripped from it and transferred over to this oxidized electron carrier, which now gains 2 electrons, each of them. Each of the 2 gains 2 electrons, therefore they're reduced to NADH. What else occurs? This right here. It requires 2 molecules of ATP to help catalyze, promote glycolysis. but you also get 4 molecules of ATP being produced. So what is the net gain? 2. So the cell gets a little bit of ATP, but in actuality, the purpose is this. Okay? Now, NADH here, which is carrying those electrons stripped from glucose, what stage did I say they're going to be taken to, everybody? I know I've been saying a whole lot. I just want to know if anybody caught it. Any electrons that are stripped from glucose or glucose-derived products. What stage? Stage 3. Yeah, stage 3. Okay? So anytime an electron carrier is reduced, that means it's gaining electrons that were either stripped from glucose or a glucose-derived product. And what is that electron carrier going to do? In its reduced form, it's going to carry them or transfer them to stage 3. You got to remember that, because that's where we're going to pick up with the first substage of stage 3. So as I said, I had some students a few semesters in a row asking me what NAD stands for. And unfortunately, every once in a while, I have a student that tests the water. So I said, okay, I'll give it to you next class. I knew where I had it. I knew where it was. So I put it up. NAD stands for nicotinamide adenine dinucleotide. This is the molecule. It is a large molecule. Okay. Am I going to ask you to recognize the molecule? Am I going to ask you to know its full name? No. I'm just showing you, okay? And it's showing you what happens when it gains 2 electrons. All right. Nicotinamide. Yeah. Along those lines. Yeah, Zachary. So if you look here, what do I want you to know for NAD? In its oxidized form, when it's not carrying electrons, we simply write it this way. This is how we let a person know it's not carrying electrons, it's oxidized. When we write it this way, now what we're letting a person know is that it's gained 2 electrons. It's in its what? Reduced form. So this is what I want you to know from this, okay? All right. Okay, this is a figure that I use for my non-majors, and I started using it for my majors, and it just gives you more of a visual to pull everything together. Remember what I said, this class builds on itself, so either it's going to get better or it's going to get worse as the semester goes, okay? So one thing you'll want to do for chapter 9 is review chapters 2 and review chapter 5, okay? Chapter 5, at least carbohydrates and proteins, monosaccharides specifically. They're showing you glucose. These little charcoal colored beads, everybody, what do they represent for this molecule of glucose? What do the little charcoal beads represent? Because it says it's glucose. Right, carbon. The lines, what do the lines represent connecting these charcoal beads that represent carbon? What do the lines represent? Right, covalent bonds, okay? So as this molecule of glucose enters glycolysis with its 6-carbon backbone, as we said, it's going to require a couple of molecules of ATP. It's going to require some energy to help the enzymes that are involved catalyze the process of oxidizing glucose, stripping electrons. But since it's primarily formed by covalent bonds, if you're going to remove electrons, you're probably going to break some covalent bonds. So what happens? This 6-carbon backbone gets snapped in half, gets broken in half, while electrons are being taken. So we go from a 6-carbon backbone glucose molecule to two 3-carbon backbone molecules. Okay, ignore the P for right now. All right, and they show you here is where our oxidized electron carrier NAD plus comes in. This is the best example I can give you for oxidized. Okay, shows you a couple of curvature areas here that represent where electrons could be carried. The oxidized electron carrier comes in and gains 2 electrons that have been taken from glucose or glucose-derived products. So it comes in oxidized and it leaves reduced. Why are they putting these minus signs in these beads, everybody? What are these minus signs representing? They're actually not minus signs. Yeah, because electrons have what type of charge? I actually have to ask this question. Negative charge. Everybody bear with me, okay? There's times I don't think I need to ask a question and then I find out, no, I should have asked it. I should have really asked it, okay? Again, review chapter 2. We talked about atoms, right? Protons, neutrons, electrons. So here they don't put an E with the negative, they just put a negative symbol in a little bead representing each of these represents an electron. You can see they've been carried. Now eventually, as these two 3-carbon backbone molecules proceed through glycolysis, eventually we get our production of a total of 4 molecules of ATP for a net gain of 2. At the end, the final product of glycolysis catalyzed by the last enzyme is 2 3-carbon backbones of molecules called pyruvate. Now listen carefully. It says pyruvic acid. Rewrite it as pyruvate. Okay, I leave it for biochemists to explain it, but you have pyruvic acid, better known as pyruvate. There's lactic acid, better known as lactate. There's acetic acid, sometimes called acetate, okay? You'll see that a lot, and I'll let biochemists explain that. But rewrite this as pyruvate instead of pyruvic acid, okay? Okay, I said there are a total of 10 enzymes that catalyze a series of 10 chemical reactions, or a chain of 10 chemical reactions, okay? You're responsible for 2. Each of these numbers, okay, represents a chemical reaction catalyzed by an enzyme. Let me ask you this. Most of the, not always, but most of the time, if a biochemist wants you to know that it's talking about an enzyme, one of the hints they give you is that they'll give you a name of an enzyme. What are the last 3 letters that it'll end in? Yeah, ACE. So notice something. Look at all these enzymes here. There are 5 enzymes that catalyze 5 chemical reactions. There's a total of 10. Therefore, 10 enzymes. But notice these enzymes, all you see is ASE. Okay? The one you need to know is this one. Okay? Well, I said glycolysis begins with the substrate glucose. Here's your glucose right here. Okay? Why does it begin with glucose? Well, main reason has to do with the enzyme. The enzyme you need to know, one of 2 for glycolysis, is the first one. Catalyzes the first chemical reaction. It's called hexokinase. Another thing biochemists will do is give an enzyme a name that kind of lets you know what the substrate or product or what it's actually doing when it's catalyzing the chemical reaction. This enzyme has a 2-part name that lets you know 2 things about what it's doing. The hexo comes from the molecule shape. What molecule shape? The shape of the substrate. Glucose has 6 sides, so what do we call any object that has 6 sides, everybody? It's not a pentagon, it's a what? Okay, thank you. All right, hexagon, okay. So the hex comes from hexagon. Well, is glucose an example of a molecule that's a hexagon? Has 6 sides? Yeah. So that's a hex part, okay? It has an active site that recognizes the shape of these 6 sides on glucose. So glucose is its substrate. The kinase, listen carefully, kinase. This is a very popular name for many different enzymes. Okay, so normally what you'll see is kinase as either being the suffix, in other words, hexokinase, or sometimes you'll have an enzyme that has a 2 or 3-part name to it, and the last part of its name is kinase. Listen carefully. Any enzyme that has in part the word kinase, that is an enzyme that phosphorylates molecules. So anytime an enzyme has at least as part of its name kinase, automatically tell yourself that's an enzyme that phosphorylates molecules. Okay, listen carefully. Phosphorylates. Phosphorylate is defined as chemically bonding a phosphate group to a molecule. So when I say phosphorylates or phosphorylation, it's defined as chemically bonding a phosphate group to a molecule. Okay. Again, it can, yes. It's just glucose preferred. So there's a little more to the enzyme that I haven't even looked at. Okay, that help you out? Yeah, because carbohydrates in general, but it's glucose. But I've never, even when I had biochem, I had it as an undergrad and grad, and I know there's more to the story, but they never said, so I never asked because I got enough to hold me over. Okay, but yeah, there's got to be more. But yes, there are other monosaccharides that can go in, but glucose definitely is the preferred one. Okay, so hexokinase, the enzyme gets its name from the shape of the substrate, glucose, and what it does to that substrate. It phosphorylates the glucose, converting glucose, the substrate, into the product glucose-6-phosphate. Listen carefully, everybody. I'm not going to test you on this, but just to let you know why the number 6. Biochemists number the carbon atoms that form the backbone. I'm not going to test you on this, but carbon number 1 is here, 2, 3, 4, 5, 6, just to let you know where the 6 came from, okay? I'm not going to test you on that, but that's where the 6 came from. So hexo, the first part of the enzyme's name, hey, I recognize a molecule with 6 sides. It happens to be glucose. So that's my substrate. It binds it at glucose at its active site. And then what does it do? It phosphorylates it. It adds a phosphate group to this molecule of glucose, attaches a phosphate group, now converting it to the product glucose-6-phosphate. Now this is the other part. Where the phosphate group comes from, notice ATP. I haven't told you this before. We're adding on to what we covered in Chapter 8. Hexokinase needs some energy. Where's it getting the energy from? ATP. It's using that source of chemical energy, ATP. And what happens though, something I didn't tell you, is hexokinase will break the covalent bond attaching the third phosphate group to the second. When that energy is released, hexokinase uses it to do its job, to do its work. But what else hexokinase does is instead of letting that third phosphate group float away, hexokinase takes that third phosphate group that it broke off and it attaches it to glucose. So sometimes you'll see this happen where an enzyme needs energy in the form of ATP, but when it breaks that covalent bond, releasing that third phosphate group, it actually doesn't let it float away. It actually goes and phosphorylates another molecule. So listen carefully. This phosphate here used to be part of ATP is what I'm saying. Okay? Now what do I want you to know? Hexokinase, substrate glucose, product glucose-6-phosphate. Does it need ATP? Yes. That's the main thing I want you to know. Now there are 10 chemical reactions. So if we go to the next slide, here's the final 6 chemical reactions that make up glycolysis. This is the second enzyme I want you to know, pyruvate kinase. Again, you can see that word kinase. That must mean that it's phosphorylating a molecule. Okay, well, what's it phosphorylating? It's phosphorylating ADP, converting it to what? ATP. So it's one of 2 enzymes responsible for producing a total of 4 molecules of ATP. Listen carefully. Look at the first part of its name. Where does the first part of its name come from? The product it produces. This is the enzyme that produces the final product, pyruvate. So is it making sense what I'm saying? I want you to know the first substrate that enters glycolysis, the final product that exits glycolysis, and then the 2 enzymes I give you, you already know part of the chemical reaction they catalyze. In this case, pyruvate kinase, the chemical reaction it catalyzes, produces the final product, pyruvate. Okay? The substrate for pyruvate kinase, it's got a long name. The substrate's name is phosphoenolpyruvate. I just want you to know it as PEP. So what'll technically happen, and I'm not going to go into this much in depth, all I want you to know for pyruvate kinase is it acts on the substrate PEP, converts it to the product pyruvate, and at the same time, technically speaking, it produces another product, which is ATP. Now, what I'm not going to test you on is how is it converting ADP to ATP? Here's that phosphate group. The kinase part of this guy takes off the phosphate group from PEP, phosphorylates ADP into ATP. There's one product. And then what's left over, minus the phosphate group, is pyruvate when you compare the 2 molecules. Okay? One thing about it, I always recommend biochem for students, but you got to make sure you have a good biochem professor, and then a good one's going to actually make you pay attention to chemical structures, and it's painful. Okay, but especially if you're going to go to med school or nursing school, I strongly recommend it. Or if you're going to go into research, okay, I strongly recommend it. But don't find one that's going to pass you along. You find one that's going to really make you pay attention to details. then that helps you understand what's going on, where names are coming from and things of that nature. Okay. So as we said down here, right, we end up with 2 molecules of pyruvate. We end up with a net gain of 2 ATP, but the purpose is this. So we end up with 2 of the electron carriers NADH in their reduced form, transferring electrons to stage 3, specifically electron transport. Now coming back up here, we are tracking what happens to 1 molecule of glucose. By the end of glycolysis, 1 molecule of glucose is converted to 2 molecules of pyruvate. What the cell does then is it transfers those 2 molecules of pyruvate to the mitochondria, from the cytosol to the mitochondria. This is single mitochondria, and ON is singular. Usually you see it plural. They drop the ON and put an A. I'm not picky about it myself. I usually just use mitochondria because that's what most of my professors did. But pyruvate will be transported into the mitochondria. When it enters the mitochondria, we end up with an intermediate step, and you need to write this down. The name of that intermediate step is called pyruvate oxidation. Well, we talked about it, right? Oxidized or oxidation, reduced or reduction. The intermediate step is actually known as pyruvate oxidation. So what is probably happening to those 2 molecules of pyruvate that are transferred to the mitochondria if the intermediate step is actually called pyruvate oxidation? What is probably going to happen to those 2 molecules of pyruvate, everybody? Yeah, they're going to lose electrons, okay? The cell, through the use of its enzymes, is trying to strip as many electrons as it can, okay, from glucose or glucose-derived products and transfer them to stage 3, okay? So listen carefully for pyruvate oxidation. You can see pyruvate oxidation requires pyruvate here in the mitochondria, and this is the final product, acetyl-CoA. So I'm going to use this step right here is this figure here, okay? It says, however, before the citric cycle can happen, pyruvate is transported into the mitochondria, okay? For obvious reasons, okay? The stage 2 and 3 happen in the mitochondria, so pyruvate has to be transferred. But pyruvate is transported to the mitochondrial matrix, and then it is oxidized, becoming acetyl-CoA. So listen carefully. It's showing you 1 molecule of pyruvate, but if we're tracking what happens to 1 molecule of glucose during cellular respiration, you would have to put ×2 for everything you see happening here, okay? But it shows you here in the cytosol, each molecule of pyruvate is transported into the mitochondria. Once the pyruvate enters the mitochondria, there is what they call a multi-enzyme complex. A bunch of enzymes that have come together as one big group, okay? And what they do first is they strip some electrons from this 3-carbon backbone. And when they do that, they break one of the covalent bonds, and that results in the release of this atom of carbon that is still covalently bonded to 2 molecules of oxygen. So in other words, when pyruvate enters the mitochondria, this multi-enzyme complex strips more electrons from pyruvate, breaking the carbon backbone, resulting in the release of CO2. There we have our first molecule of CO2. Down here, the 2 electrons that are stripped from pyruvate, well, this multi-enzyme complex strips them from this pyruvate and turns around and hands them over to the oxidized electron carrier NAD+. It gains 2 electrons, therefore it is reduced to NADH. And again, where is it going to take the electrons, everybody? Stage what? Please give me an answer. Yeah, stage 3. Stage 3, electron transport. But this is what else happens. It's not only being oxidized, technically a molecule is being attached to it by this same enzyme complex. A molecule called coenzyme A, better known as CoA, will be covalently bonded to the remaining 2 atoms of carbon that form the backbone. So in other words, what's happening is this enzyme complex is swapping off these 3 atoms for this molecule. Notice how this is replaced with this while at the same time oxidizing it. So pyruvate oxidation is occurring. We have this molecule called coenzyme A or CoA added, and now we have our final product, acetyl-CoA. Listen carefully. The final product of this intermediate step, the final product of pyruvate oxidation is acetyl-CoA, okay? But acetyl-CoA is the substrate, the starting substrate or the beginning substrate needed for the citric acid cycle. So acetyl-CoA is the product produced by this process, but then it becomes the first or the initial substrate needed to begin the citric acid cycle. Okay? So when you look at this here, there's glucose and then pyruvate and acetyl-CoA. Where did they come from initially? Glucose. So this is what I mean by glucose-derived product. Here's 2 examples. Notice the arrows keep going straight across. Notice with glycolysis, they show you a substrate and then a final product. Then they show you a straight arrow because pyruvate now becomes a substrate converted to a final product. But when we talk about this on Wednesday, acetyl-CoA, okay, this is the beginning, the starter substrate, the beginning substrate needed for the citric acid cycle. But notice, on Wednesday, I won't mention any products with the citric acid cycle. It just goes in a circle. When we get to stage 2 on Wednesday also, now we not only have the electron carrier in its reduced form NADH leaving glycolysis and this second intermediate, or in this intermediate step, but we're going to talk about another electron carrier that gets involved. So the citric acid cycle, its job is to complete the process of oxidizing glucose, or more specifically, oxidizing glucose-derived products. In other words, a whole lot more electrons are going to be stripped. So there's actually no product left, is what they say. It just keeps going in a cycle. Okay. All right, are there any questions? This is why I wanted to break it up. At first, I was like, oh, I went too fast through 7, but then it occurred to me, no, there's no easy chapter. Okay, I'm going to stop the recording.